Hello everybody, welcome back. We are going to tackle chapter 6 today. This is going to be part 3 of our unit 1 lecture. Chapter 6 is going to cover 3 main topics. We're going to take a look at extranuclear inheritance, which covers DNA that is not found in the nucleus, but in other organelles such as the mitochondria and the chloroplast. We're also going to take a look at epigenetic inheritance and imprinting. This is going to cover if there's any alterations to the gene after the production of offspring. And then a very interesting topic that we're also going to discuss in chapter 6 is this thing called maternal effect. Maternal effect is going to discuss the fact that the mother will determine the trait of the offspring regardless of what the contribution of the father happens to be. All right, so let's start with our first one, which is extranuclear inheritance. And thus far, we've been talking a lot about DNA, and hopefully by now, we can safely summarize that our DNA is our genotype, and within that, we have our genes, which are pieces of DNA that are able to do transcription and translation. And in essence, the genotype, along with environmental features, is what ultimately will determine our phenotype, our physical attributes, our behavioral traits, genetic markers, diseases, etc. and etc. Now, in chapter 6.1, what it talks about is the fact that there are other places in our cells that we have DNA. Now, for us, it's going to involve looking at the mitochondria and its mitochondrial DNA. But when you look at plant and cyanobacteria, you also see that they have DNA within the chloroplast organelle. Both the mitochondria and chloroplasts are going to be an example of cytoplasmic inheritance, meaning that you have DNA that comes from an extranuclear inheritance, so outside of the nucleus, that is then donated from parent to offspring. And what's interesting about the DNA that we're going to find both in the chloroplast and the mitochondria is that they're both going to be a single circular double-stranded DNA. And usually what we see happening is that within these organelles, the chloroplast and the mitochondria, the DNA will be housed in what we call a nucleoid. Now, a nucleoid is not to be confused with a nucleus, because a nucleus is the control center of the cell. The nucleoid, on the other hand, is more of a housing or a storage unit. It is very similar, or it is the same as what prokaryotic cells such as bacteria utilize to house their DNA as well. So in this particular slide, it just discusses the fact that chloroplast, which is what we see in plant and cyanobacteria species, does indeed has its own DNA, and that DNA is going to be in a singular, circular, double-stranded format, and is going to be housed in nucleoids. And it's actually quite common that within different organisms, the chloroplast will have multiple copies of its DNA within the nucleoid. And there's also been some cases where there'll be multiple nucleoids in the chloroplast as well. So there's lots of options for that. The DNA that's in the mitochondria, as I was mentioning to before, is also going to be singular circular chromosomes, and very similar to the chloroplast, it's going to be housed within the nucleoid. Now, something I want you to think about is the fact that in eukaryotic cells, we see that the nucleus houses our nuclear DNA, which is the DNA that's inherited from both maternal and paternal contributions. And that DNA will then go ahead and have sections that can do transcription and translations, which also involve recruiting other organelles and in essence coming together for the cell to have its overall function. So the other organelles that you find normally in the cytoplasm of a eukaryotic cell, things like smooth ER, rough ER, the Golgi apparatus, the vacuoles, the peroxisomes, they all depend on the DNA provided by the nucleus, in essence, for their sense of directions. So what happens with the DNA that we have in our mitochondria? Are we utilizing it? Are we ignoring it? Well, it turns out that within this one single circular chromosome, there's about 13 genes of interest, And a lot of them will have to do with allowing the mitochondria to do oxidative phosphorylation, which is, its main concept of being able to convert energy into ATP. Most of the time, though, the DNA from the nucleus will encode proteins that can directly communicate with the mitochondria, thereby allowing it to send signals and have targets as to what the mitochondria needs to do. So we see in a lot of eukaryotic models, also in some prokaryotic models, that if there is the presence of the mitochondria, that the nucleus does have a sense of communication with that organelle. Something that I definitely want to point out about the mitochondrial DNA, the DNA that you find in your mitochondria, is that its only contributor is from your mother. It is often called mitochondrial DNA or maternal DNA because your mother is the only one that will contribute to it. So that means, for instance, that if you've ever watched, shows where they do DNA analysis to determine the father of a child, when they do analysis like that, they go over to the DNA that's inside of the nucleus because we know that comes from mom and dad. On the other hand, if there is a case, for instance, where there maybe is like a missing child and the mother steps forward to say, hey, that's my biological child, what we see happening is that they would extract the DNA out of the mitochondria. And if that mother is indeed biologically related to the child, it would be 100% match compared to the DNA that she has in her mitochondria. Now, hopefully, as we're kind of chit-chatting, or as I'm kind of chit-chatting about the DNA that you have in the chloroplast and the mitochondria and how that's unique when you take a look at your other organelles, some of you are going, well, how exactly did the DNA get there? What is our proof? Like, what is happening? Why can't the nucleus just take care of all the DNA that you house? And it turns out that after extensive research, we can explain it by the endosymbiosis theory. So the endosymbiosis theory basically states that billions of years ago, we see that the chloroplast was originally a cyanobacteria, and the mitochondria was originally a purple bacteria, most likely from the gram-negative category. And somewhere along the way, both of these individual entities, so the cyanobacteria and the purple bacteria, were engulfed by a larger cell. And instead of that cell then digesting the cyanobacteria and the purple bacteria, we see that the cell became a sort of host and incorporated these organisms into its own genome. And every single time the cell would replicate, so would the cyanobacteria and the purple bacteria. And over time, we see that both of these then independently evolved. The cyanobacteria went ahead and evolved into what's now called the chloroplast, which allows things like photosynthesis to occur within the cell, whereas the mitochondria was the end product of the gram-negative purple bacteria that was engulfed by the host cell. And without this mitochondria, we would by no means be able to generate as much chemical energy in the form of ATP as we are able to do so. In the illustration that is on our right-hand side, you can go ahead and see that when we take a look at plant and algae models, we're basically going to utilize the endosymbiosis theory twice because they have access to both the chloroplast, which was originally the cyanobacteria, and the mitochondria, which originally was our purple bacteria. In other animal and fungi models, what we see is that they do not photosynthesize. So in that case, what we see happening is that endosymbiosis only had to occur once to allow for the incorporation of the mitochondria. I like to think of it kind of like Pac-Man. The larger cell ate the smaller cell and incorporated it into its genome. And I know it sounds kind of weird, but there is specific evidence that they have that can prove this endosymbiosis theory. And the evidence is the fact that the chloroplast and the mitochondria still till to this day have their own DNA. So that's proof that they were able to survive independently. But also when we take a look at their structure, we see that they have a double membrane. So when you look at the chloroplast and the mitochondria, we see that they have a double membrane that encases the outside region. It is a double membrane because the internal membrane belonged to the original cyanobacteria and the purple bacteria, and the secondary membrane that was placed on top of the internal one, that was placed there by the host cell, declaring it that it now belonged to its own genome. So those are the two pieces of evidence, the fact that both have their own DNA and they have a double membrane, whereas all the other organelles that you would find in your eukaryotic cells only have a single membrane and they do not have their own DNA, with the exclusion, of course, of the nucleus, thereby showing that the chloroplast and the mitochondria once upon a time were free living, independent organisms. The next item in Chapter 6 is to take a look at epigenetic inheritance. Epigenetic inheritance is sometimes also called genomic imprinting. It's when we see that there's a modification to the gene that allows the gene expression. Oftentimes these modifications tend not to be permanent. They can be permanent throughout the life of the individual. However, when it's time for the gametes to form, we see that the modifications might be lost. There tend to not be an actual change to the DNA sequence itself. For instance, when we were talking in our previous chapter about X chromosome inactivation, that's a perfect example of modifying a gene that will ultimately cause a change in your gene expression. And we now know, obviously, that the X chromosome inactivation is done completely at random, and we see that once the gametes are produced, both X chromosomes become fully active, because as we can see, that is then when the cell goes from diploid to haploid. If you've ever heard of DNA methylation, when the methyl is added to the cytosine in DNA, that would be another example of epigenetic inheritance. And obviously, as I just mentioned to you, genomic imprinting is another example that falls in this category. Genomic imprinting, though, is a little bit of a more specific example, because in that case, what you look for is you look for an expression of a gene, depending on if it's inherited from the male or the female parent. So here we go. So here is our full definition of genomic imprinting. The expression of a gene depends on whether it's inherited from the male or the female parent. This obviously goes completely against what Mendel was predicting. He basically said that whoever gave the dominant gene would have the dominant phenotype being expressed. And genomic imprinting says, nope, it depends on what mom or what dad is contributing to the fact and which one we are going to be listening to or which one we are going to be expressing. So in genomic imprinting, since it's either parents could be the possibility as to which one we're looking at, we can also go ahead and call it monoallelic expression, meaning that you only express one allele regardless of what the other is contributing to the system. Let me go ahead and show you a picture with a nice example of genomic imprinting. Okay, so here we go. So we are going to go ahead and take a look at what we like to call the insulin-like growth factor, IGF. And the way it's going to work with this particular gene is that if IGF-2 is indicated with a little negative sign, like right over here, that means that you have a mutation that causes a loss of function. And this particular mutation will go ahead and affect the overall growth of our mouse species. So here we have a little mouse. And what we see happening is that if there's a mutation and you have a loss of function of the insulin growth factors 2, we see that the offspring will often be termed as dwarfs, in that they will be a lot smaller than the wild type offspring. So here's the normal size, and then here we have the ones that are knocked out. So they have the negative attached to it. And what we're going to do here in this example is we're going to take a look at which parent we're going to listen to, because we're talking about genomic imprinting, which basically says that we're only going to focus on the expression of the gene from one parent, regardless of what the other is going to do. And in fact, in our particular example, it's going to turn out that we are going to follow the father's lead, and we're going to go ahead and ignore what mom is contributing to the signal. So in essence, we are going to transcriptionally silence mom's genes. And let me show you what I mean with that. Okay, so let's take a look at our two examples, right? So Punnett square 1, here you go, nicely highlighted. You can see that mom is homozygous for the knockout, and you can see that father is homozygous for the wild type. So here we have all the genes that would carry for the dwarfs, and then here you would have the gene that would come in for the normal size. Now, I know by now we have done a few Punnett squares already, but as you can imagine, since both parents are homozygous, one homozygous for the wild type, one homozygous for the mutation, what we see happening is no matter how we do our Punnett square, all offspring will become a heterozygous mix, one representing the knockout and the other one representing the wild type. Now, look what's going to happen though. You would assume that since you had one wild type and one knockout, that you would have some type of abnormalities that would come into play. But it turns out that what we're going to be doing is we're completely going to be silencing any of the alleles that come from mom. So that means that all those alleles that came from mom, that were the knockout, that were encoding for the dwarfs, we're just going to ignore it, even though it's still there. And the fact that it's still there and that we're ignoring the mother's contribution is an example of genomic imprintation. And what we see happening is, since we're only going to follow what the father has contributed, that means that when we take a look at our somatic cells in our heterozygous offspring, all of them are going to go ahead and be completely normal. No signs of abnormal growth are going to be detected. Once again, we're doing genomic imprinting, where we're just following what the father has contributed to the Punnett square. Now, our second scenario, we're going to go ahead and we're going to flip it. And what we're doing now is we're going to take our homozygous wild type and give that to mom, and we're going to take our homozygous knockout loss of function gene, and we're going to go ahead and give that to dad. And once again, you're going to produce offspring that are heterozygous. They have one of each. And what do we see happening here? Once again, we're going to ignore what mother is contributing, which means that in this case, we're only listening to the knockout gene inherited from dad, which means that all offspring will be labeled as dwarfs, a lot smaller than the wild type. Once again, we're doing genomic imprintation, where we are ignoring any other contribution except for the one that the father has done. So I kind of established this before, but what we're doing is we're basically transcriptionally silencing the maternal lineage. And in case you're wondering, well, is it always the father? No, there's been plenty of cases where we will go ahead and only listen to the maternal lineage and we'll ignore the paternal lineage. We also see that which parent is going to be the expressive one is usually established during gametogenesis. And then once it's decided which one will have the lead, that decision will stay throughout the embryonic development and somatic cell development. And once the offspring is ready to start producing their own gametes, we often see that the choice will go ahead and reset itself, and then it can be fair game as to whether you will be listening to the maternal or the paternal contribution. All right, in fact, if we're looking at models where only the mother has an effect on the expression of the offspring, we're going to call that a maternal effect inheritance. And in a maternal effect inheritance, we're basically going to ignore anything that the father contributes, so the genotype has absolutely no effect, nor will the actual genotype of the offspring. This is all going to be under control of our nuclear genes, and it turns out that after research, it's the maternal effect is due to the fact of the different nurse cells and diploid cells that the mother will be utilizing to, in essence, produce and nourish her oocyte. And we're going to talk more about that and her developing eggs and their haploid ability in a little bit. But for now, let's go ahead and take a little example, look at an example of the maternal effect. And keep in mind, in our example, we are only caring about the genotype of the mother. So the genotype of mom will determine what the offspring will look or behave like. Okay, so here's our little example, and what we're going to do is we're going to go ahead and take a look at water snails, and we're going to take a look at the orientation of their shell, and we basically have two options. We have what we call dextral and sinistral. Dextral is indicated by the capital D because that's our dominant trait. And dextral means that when you look at the axis and the coiling of the snail, it's going to be more towards the right. So if you see it coiling toward the right, it's a dextral trait, and that is the dominant trait indicated by the capital D. The lowercase d is going to be the recessive trait. That is going to be coiling towards the left, which we like to refer to as sinistral. So sinistral is the lowercase d. That means that we're moving towards the left. And dextral is the capital D. That means that we are coiling towards the right. Okay, so let's go ahead and take a look at this particular reciprocal cross. And what we see happening here is that in our first scenario, we see that mom is homozygous dominant, correct? Dad is homozygous recessive, and as you would predict when you do your Punnett square, since both of them were homozygous, all of the offspring is going to be heterozygous. So far, so good, right? Because mom gave a capital D, dad gave a lowercase d. All the kids are going to be heterozygous. Now, it turns out that all the kids will go ahead from a phenotypical aspect. They'll be dextral. Now, some of you are going, but isn't that what we are expecting? Yes, we're expecting that according to Mendel's law, okay, but since we're discussing the maternal effect, we really also want to double check that the dextral would be the phenotype based on the genotype that was inherited from mom. And in this case, in our first example, it makes complete sense because mom was fully homozygous dominant, which means that she was dextral herself, so that would make sense that her offspring are dextral as well. In fact, let's go ahead and go over to our reciprocal cross right next to it, and let's take a look at the flip scenario. So in our second scenario, we see that mom is going to be homozygous recessive, right? So she's coiling to the left, she's sinistral, and dad is going to be homozygous dominant. And once again, we do our Punnett square, and we see that all of our offsprings are going to be heterozygous. Now look what's going to happen over here. We would expect that the dominant trait would show through, right? Because that's what Mendel is telling us. Now read the sentence below right here. It says all offspring is sinistral. But wait a minute, sinistral is the recessive trait. That's when you have the lowercase d. But we have a capital D, we have a dominant D. So why are we ignoring the dominant gene? Well, because we're looking at an example of the maternal effect where it doesn't matter what dad brought to the table. This is me wiping out dad. Sorry, dad. We only care about the genotype that mom is contributing. And since mom was fully dextral, was fully sinistral, excuse me, she was homozygous sinistral, that means that all her offspring will be sinistral as well. So whatever mom gives is what the offspring is going to be, regardless of what dad is contributing to the system. And in fact, what we see happening is if we go ahead and we do another Punnett square where we're going to do our two heterozygous mixes together, it turns out that when we plug it in, instead of getting our regular prediction of 3:1, we see that all the offspring end up being dextral. And I'm hoping you're going, wait, what? How are they all going to be dextral? Well, remember, if you're doing your Punnett square here, right, mom is going to be heterozygous, and dad is going to be heterozygous. Because at the end of the day, both parents had the same genotype. Now, if mom is heterozygous, that means that the expression of the gene that we're going to be looking at is going to be the dextral gene, because mom is going to be dextral. So if you do your Punnett square, you're going to see that you do indeed end up with one offspring that's homozygous dominant, two offspring that's heterozygous, and one offspring that's homozygous recessive. And because you're expecting the 1:2 ratio with the dominant gene, that is where the 3:1 predicament comes in for the Mendelian pattern. However, remember, we don't care what dad is bringing to the system. The fact that mom had a capital D, that means that the dextral trait is the one that will be showing through, which means that we really don't care what dad is contributing. All offspring will be dextral. And this trend will continue. So if you go ahead and now take your 3 different scenarios and you go ahead and you start doing crossing them with each other, we see that there are scenarios where mom will be homozygous dominant and heterozygous. So any offspring produced from this scenario, these 2 scenarios, will go ahead and be dextral. And if we do a cross where mom is homozygous recessive, then what we see happening is that her offspring from that particular trait will go ahead and be sinistral, because that is once again the genotype that mom is passing along to her offspring. So here we can go ahead and we can take another look at it. So we started off with our parental generations, where we were looking at homozygous dominant and homozygous recessive. We're creating an F1 generation that's heterozygous, where we would expect the dominant gene to be the permanent one or the more expressive one. But what we see happening in our F1 generation is that we do not care what what father is contributing. We're only looking at what the mother is contributing. So in this case, in our first scenario, since mom is dextral, all offspring is dextral. And if mom is all sinistral, then all offspring is sinistral. And we continue this trend line by now taking the heterozygous genotype and going and crossing them against each other to create our F2 generation. And when we do that, what we see happening is that we start seeing some dextral organisms pop up in the F2, where some of you might be expecting some sinistral. Now remember, in the F2 generation, all the offspring will be dextral, because when you do your cross, the mother is heterozygous. And the fact that she's heterozygous means that she is giving her dominant gene, her dextral gene, to her offspring, regardless of what the father is bringing to the party. Therefore, all the phenotypes in F2 are going to be dextral. And we see that trend continue on with our F3 generation, where now we're doing a cross, where some of the mothers have the capital D in either homozygous or heterozygous form, thereby allowing for the dextral offspring to be produced. But we also have the scenario where you have your homozygous recessive trait, and if mom is donating her sinistral gene and her sinistral genotype to the offspring, guess what they're going to be? They're going to be sinistral. So in the maternal effect, you really only care about the genotype that the mother is contributing to the offspring. We are literally just ignoring what the father is contributing, and sometimes we're also ignoring the Mendelian genotype or Mendelian prediction that the offspring would potentially be. And it turns out this particular non-Mendelian inheritance pattern is seen during the process of oogenesis. And it all comes from the fact that the individual eggs, as they're being nourished and developed, are surrounded by maternal or nurse cells. These nurse cells are diploid, and they are able to produce proteins that go ahead and are introduced into the oocyte and will contribute to their expression of particular phenotypes. Now go ahead and take a look at the sentence right here, and it just reminds you that, remember, the nurse cells or the maternal cells that take care of the oocyte are going to be diploid. 2 copies of everything, whereas the developing oocyte, the egg, is going to be haploid, 1 copy of everything. And what I want to do on the next page is I want to show you how the nurse cell will go ahead and control the expression of the gene in the oocyte. And we can utilize that example to go ahead and explain some of the non-mandibular patterns that we saw in our example when we were looking at the crosses for the snail. Alrighty, so here we go. So here is our oocyte. So here you go. There's your little oocyte. And what we see happening is that we have diploid cells surrounding it. These are going to be your maternal cells or your nurse cells. And what we're seeing happening in our maternal effect is that the proteins that are being generated by the genes of the nurse cells will have a full effect on the expression of the phenotype of the actual egg. So let me go ahead and open the box up so you can kind of see it. And basically what we see happening here is that the gene product from the nurse cells will basically persist throughout after fertilization, allowing the oocyte to express the genotype of the parent, of the mother, I should say, ignoring what contribution the father has done. So here are our 3 scenarios when we take a look at the maternal effect, okay? So you can have a mom that's homozygous dominant, meaning that she has nothing but dextral genes that are being expressed. You can have a mom that's heterozygous, meaning that she's able to produce an egg that's either dextral or sinistral. And you can have a mom that's homozygous recessive, which means that no matter how you twist or turn it, all eggs are going to go ahead and be sinistral, carrying the recessive trait. All right, now what we're going to see happening is, depending on the dominant proteins that the nurse cells generate, they will contribute to the gene product that the egg will place on the expression of the offspring. So what do I mean with that? Well, in our first example, we see that our nurse cells are only producing the protein for the dextral coiling, or the dextral coiling of the snake's, the snail coil, excuse me, meaning that everything is pointing towards the right-hand side. So what we see happening is that all the gene products will allow the egg to express the D allele. So all the offspring that's being produced by this mother who's homozygous dominant, all the offsprings will go ahead and become dextral, regardless of what the father is contributing. Okay? Now, let's go ahead and skip to the last one right here. In this case, we're looking at a homozygous recessive trait. When we're looking at our nurse cells, we see that all the nurse cells are able to generate the protein for our lowercase d, which means that it's the expression of the sinistral trait, which means that that is the only expression of protein that we see from the maternal end, which means that all offspring that come from this particular mother, all offspring is going to be sinistral, because that is the protein that was inherited or passed along from the nurse cell into the oocyte. So that then leaves our last one, which is our middle one right here. And this one basically says, if the mother is heterozygous, that means that the egg can either do a capital D for the dextral or a lowercase d for a sinistral, right? But what's interesting to see here is that the nurse cells are following the Mendelian trait of expressing the protein for dextral at high levels. And because they are expressing the protein of the dextral at such high levels, what we see happening is that, very similar to our first 2 examples, this protein is fed into the oocyte, and as a result, it will go ahead and cause any offspring that comes from the mother that is heterozygous, all offspring will be dextral. And the reason for that is because they are responding to the dominant D allele and the capital D protein that's being generated by the nurse cells. So once again, we are completely ignoring what the father is contributing to the system. We are only looking at the gene product that the mother is introducing, and it will then completely control the expression of the offspring. So please do me a favor and go ahead and go back to this particular slide, and I want you to kind of talk yourself through it and see if you really understand what's happening. And keep an eye on the fact that you are doing the maternal effect. You are only looking at the genotype that the mother is passing along. We do not care what the father is doing. And as you work your examples through it, you want to kind of ask yourself, what part of it is following Mendel's law and which part of it is deviating from its expression. All right, and please keep in mind that as we migrate from our F1 to our F2 generation, that you're looking at a genotype for mom that's heterozygous, right? And that means, as we were looking at our previous slide, that the nurse cells are producing that protein for D at extremely high levels, which means that from a phenotypical aspect, all offspring will be dextral. The only way that you can generate offspring that will be sinistral is if you're looking at an example where the mother is homozygous recessive, so she has two of the lowercase d's that she's going into the system with, because in that case, the only protein that the nurse cells are generating are going to be for the sinistral coiling. And on this little picture right here just kind of shows you how the mitotic spindle either will lean towards the right or towards the left, and due to that orientation, it will cause the coiling of the snail shell to either be dextral to the right or sinistral to the left. So for those of us who are a little bit more visual and want to see which way we're going, this just kind of shows you how the mitotic spindle will guide the coils. Alrighty, so over here, just kind of finishing off this particular part of the maternal effect. Keep in mind that the maternal effect genes encode RNA and proteins that play an important role in the early steps of embryogenesis. And this was basically taking a look at the example of what the nurse cells were able to contribute to the developing oocyte. Let's see what else it says. It says accumulation before fertilization allows development to proceed very quickly after fertilization. Therefore, defective alleles in maternal effect genes tend to have a dramatic effect on the phenotype of the individuals. There's also been work done on Drosophila, so fruit flies, in addition to the snail coil, to see what would happen if there are some of these mutations present in the mother's nurse cells or the oocyte, how that would potentially affect the overall appearance and behavior of the offspring. Now guys, this part is a nice little short lecture, so I want you to kind of work your way through it, because on your exam there will be several questions about the maternal effect, and I will most likely also ask you some offspring questions. So if I give you a scenario where mom is this genotype and dad is this genotype and we're doing the maternal effect, what will that mean? So I really want you to take your time and walk yourself through my explanation, and in case it wasn't clear, I don't take offense to it. By all means, go ahead and send me that email, post it in the discussion session, and let's talk it out, because I really want you to get a nice understanding of this maternal effect, and obviously also the other two subjects of Chapter 6, which was your genomic imprinting and the extranuclear inheritance that we kind of chit-chatted about. But definitely the most important part of this particular audio lecture is going to be the maternal effect. Alrighty, we will talk soon again, and whenever you're ready, I'll see you for the end part of part 3, which will actually also conclude our Unit 1 lecture. Talk soon or listen soon. Bye.